Lecture 1: Biogeography and Soil Biology. Let us begin our exploration of general biology with biogeography, the study of the distribution of organisms across the Earth and the factors that influence their locations. It is a field that connects ecology, evolution, climate science, and geography. Globe showing Europe-Africa 1.1 distribution of world flora and zoogeographic regions. The Earth's biodiversity is not evenly distributed. Instead, it is influenced by climate zones, geological history, and barriers such as oceans and mountains. Floristic regions include O boreal, northern forest. O paleotropical, Africa and parts of Asia. O neotropical, South America. O Australian and Antarctic. O South African, Cape Flora. Each region hosts distinct plant communities shaped by evolution and adaptation. Zoogeographic regions include O Nearctic, North America. O Palearctic, Europe and Northern Asia. O Afrotropical. O Indomalayan. O Australasian. O Neotropical. O Oceanic. These regions reflect where animal species evolved and migrated, often in relation to plant availability. Desert Island 1.2 Island Biogeography and Dispersal. Island biogeography is a key concept in understanding how species colonize new territories. It emphasizes the distance of the island from the mainland, affecting immigration rate. The size of the island, affecting extinction rate. Species richness as a balance between immigration and extinction. Dispersal mechanisms include wind, water currents, animal-mediated transport, human activity. These mechanisms help plants and animals reach and colonize new habitats, especially isolated islands. Seedling 1.3 Vegetation, soil types, and climate. Plant distribution is deeply connected to climate and soil. For example, tropical rainforests thrive in nutrient-poor but moist soils with year-round warmth. Temperate forests grow in well-drained, seasonally varied soils. Deserts host drought-tolerant vegetation in sandy, arid soils. There is a mutual relationship between vegetation and soil. Plants influence organic matter content, erosion, and water retention. Soils provide nutrients and structure for plant growth. Test tube 1.4 Soil classification and analysis. Soils are classified based on texture, sand, silt, clay. Structure, color, pH, nutrient content. Soil analysis includes pH testing, organic matter content, nutrient testing, e.g., nitrogen, phosphorus, potassium, moisture content. Soil types influence crop suitability, microbial activity, and even animal distribution. Basket 1.5 Soil sampling techniques. In local habitats, we use several sampling techniques. Random sampling with quadrats. Core sampling for deep layers. Transect lines for gradient analysis. Augers for compact soil. These help identify soil properties and biodiversity at various depths. 1.6 Adaptations to subterranean life. Organisms living underground, like moles, earthworms, and bacteria, show unique adaptations. Reduced eyesight. Increased tactile or olfactory senses. Streamlined bodies for burrowing. Symbiotic relationships with plant roots. These life forms play crucial roles in aeration, decomposition, and nutrient cycling. Studio Microphone Lecture 2, Microbial and Molecular Biology. Microscope 2.1 Microbial Genetics. Microorganisms like bacteria, archaea, and fungi are widely used in genetic studies due to their short life cycles, simple genomes, rapid reproduction. They help us understand mutation, gene regulation, and recombination. 2.2 Genes, chromosomes, and nucleic acids. Genes are units of heredity made of DNA. DNA is structured in double helices composed of nucleotides A, T, G, C. Prokaryotes usually have a single secular chromosome. Eukaryotes have multiple linear chromosomes in a nucleus. DNA contains coding regions, genes, and non-coding sequences, regulatory elements. 2.3 DNA replication, transcription, and translation. Replication, the DNA makes a copy of itself. Transcription, DNA is transcribed into messenger RNA, mRNA. Translation, mRNA is decoded into a protein at the ribosome using tRNA. This flow of information is known as the central dogma of molecular biology. Gear 2.4 Gene expression and regulation. Not all genes are active all the time. Regulation is influenced by promoters and enhancers, transcription factors, environmental signals, e.g., nutrient availability. In bacteria, operons like the lac operon provide models for gene regulation. Petri dish 2.5 Gene sequencing and protein synthesis. Gene sequencing identifies the order of nucleotides in DNA. Technologies include Sanger sequencing, next-generation sequencing. Protein synthesis involves initiation, elongation, and termination stages. Amino acids joined by peptide bonds. Enzymes folding into complex shapes for function. This field has major applications in genetic engineering, medicine, and agriculture. Studio Microphone Lecture 3, Algae. Algae are photosynthetic organisms found in marine and freshwater environments. Though often grouped with plants, many algae are protist or even bacteria, e.g., cyanobacteria. Petri dish 3.1 Systematics and morphology. Algae are classified into major groups. Green algae, Chlorophyta. Brown algae, Phaeophyta. Red algae, Rhodophyta. Diatoms. Dinoflagellates. They exhibit varying morphologies. Unicellular, e.g., Chlamydomonas. Colonial, e.g., Volvox. Filamentous, e.g., Sparogyra. Thalloid, e.g., Laminaria. Waterwave 3.2 Reproductive systems and life histories. Algae reproduce asexually, binary fission, fragmentation, spore formation. Sexually, isogamy, anisogamy, oogamy. Many have alternation of generations, switching between haploid and diploid stages. Seedling 3.3 Ecological and economic importance. Ecologically, algae are primary producers in aquatic ecosystems. They produce oxygen and form the base of the food web. Economically, alginates from brown algae are used in food and cosmetics. Agar and carrageenan from red algae serve in microbiology and food. Algae are being explored for biofuel production. Studio Microphone Lecture 4, Mycology. Mycology is the study of fungi, which are eukaryotic organisms distinct from plants and animals. Mushroom 4.1 Systematics and morphology. Fungi include molds, e.g., Rhizopus. Yeast, e.g., Saccharomyces. Mushrooms, e.g., Agaricus. They have hyphae, thread-like structures forming the mycelium. Cell walls made of chitin. No chlorophyll, they are heterotrophic. Counterclockwise arrows button 4.2 Life cycles and dissemination. Fungi reproduce asexually, spores, budding. Sexually, fusion of hyphae, meiosis. Dissemination occurs via airborne spores. Water. Animals and insects. Fungal life cycles vary. Zygomycota, bread molds. Ascomycota, yeast, truffles. Basidiomycota, mushrooms, rust. Lightbulb 4.3 Economic importance and mycological techniques. Fungi are pathogenic, cause diseases in plants, animals, and humans. Decomposers, recycle nutrients in ecosystems. Beneficial, produce antibiotics, e.g., penicillin, alcohol, via yeast, and food, e.g., mushrooms. Mycological techniques include isolation on culture media, e.g., PDA. Microscopy, staining spores, hyphostructures. DNA sequencing for classification. White Heavy Checkmark Module 1 Summary. In this module, we explored 1. Biogeography, global distribution of plants and animals, island colonization, and soil-plant relationships. 2. Soil biology, soil classification, sampling, and adaptations to underground life. 3. Microbial and molecular biology, genes, replication, protein synthesis, and gene regulation. 4. Algae, diversity, life cycles, and ecological roles. 5. Mycology, classification, reproduction, and economic significance of fungi.